So today we'll be looking at the characteristics of enzymes. What makes an enzyme to be an enzyme, or what makes enzymes to be enzymes? That is what we are going to be looking into, right? So the first thing I have on the board here is that enzymes act as catalysts which help to speed up or slow down the rate of most chemical reactions within the body of the organism. That is to tell you that enzymes are organic catalysts. I already said that at the beginning of this course while I was explaining the concepts of enzymes. So you just have to note that enzymes are catalysts. And what does a catalyst do in a reaction? The catalyst basically speeds up the rate of the chemical reaction without altering the form of that particular compound. And now in the context of the enzyme, what it does is that it comes into the reaction to speed up that reaction by reducing the what we call the transition state, or the activation energy of that complex. So that is for that. Going forward, I'm still going to explain how these enzymes does this. So for the number 2, we say that they remain chemically unchanged at the end of the reaction. I already said that while I was trying to explain the first characteristics. So enzymes do not get used up in a chemical reaction. Thirdly, I said enzymes are specific in action. That is, one or a group of enzymes will act on specific substrates. For example, the thiolane in the saliva acts on courstar, changing the good starch to maltose sugar. What this simply means is that every enzyme is peculiar to a type of reaction. That is to say, one enzyme or two enzymes cannot catalyze the same reaction at the same time or at the same location. Although we have what is known as isozymes, which is already written here on the board. Isozymes are basically two enzymes that perform the same catalytic function at different locations of the body. For example, we have the glucokinase. We have the glucokinase and the hexokinase. Right? These two enzymes are known as isozymes. What do? Glucokinase and hexokinase and hexokinase are isozymes. What do? Glucokinase is responsible for the phosphorylation of glucose. That is the first step of the glycolytic pathway. Don't worry, you're going to do this when you get into the higher institution. Those of you that will be doing glycolysis, you're going to understand it better when you get there. So I'm just using this to illustrate what isozymes are. So the glucokinase catalyzes the phosphorylation of glucose to glucose 6-phosphate in the liver, while hexokinase is responsible for the phosphorylation of glucose to glucose 6-phosphate also, but this guy acts in the muscle cells and other part of the body, while the glucokinase itself is found in the liver. Right? That is it. So the fourth example, or the fourth characteristics of enzyme is that they are required in small quantity. Right? Enzymes are required in small quantity. When you want to do your analysis in the lab and you're making use of an enzyme, you're basically going to use a very small quantity of the enzyme of interest in your analysis. You don't have to go through the stress of getting a lot of sample of that enzyme to do your work. So they are basically required in small quantity. And I went further to say a single of one particular enzyme can change 1,600 molecules of food in a second. That is to tell you how swift and how powerful they are in their function. And I also said in the fifth characteristics of enzyme that enzymes are pH specific. What it simply means is that some enzymes have the ability or the capacity to operate under an acidic medium or an alkaline medium. Because in your pH scale, you have the acidic part and the alkaline part. In the pH scale is just a scale that operates from 0 down to 14, right? And in between, there's a neutral point, which is 0.7. So from 0 to 7 is acidic, and then from 7.1 down to 14 is alkaline, right? So that is to say, those enzymes that will act on the acidic medium will have the ability to catalyze reactions under an acidic environment. For instance, in our stomach, the pH of the stomach is actually an acidic condition. So there are some enzymes that will act freely on our stomach, while there are some, like those like in the small intestine now, which is an alkaline condition. Some enzymes will also act on that alkaline medium. So that is to tell you that some of these enzymes operate very well at acidic condition, while some of them operate at an alkaline condition, right? And in the sixth characteristics, I said most enzyme actions are reversible. Most enzyme actions are reversible. What does that simply mean? It simply means that the enzyme, a particular enzyme, can catalyze a reaction to give you a product. That product can be also acted upon to give you back the initial substrate that gave that product. I know it might sound confusing, but it is very simple. If you look at this equation, I have here on the board, it is a schematic representation of an enzymatic equation, right? So this E here stands for the enzyme of interest, right? And the S here stands for the substrate. Now, what is a substrate? The substrate is simply that compound which the enzyme is going to act on. We refer to them as substrates. Now, the reaction between the enzyme and the substrate leads to the production of an ES complex, which is known as the enzyme substrate complex. The enzyme substrate complex, which now leads to the formation of the product and then the enzyme, which is unchanged or unused in the reaction. Now, if you look closely, on getting to this ES complex is more like an intermediate of the reaction. We can move from the ES complex back to the enzyme itself and the substrate itself, which goes to tell you that this reaction can be reversed. That is why we are having these two arrows, one going this way and the other one going this way, which is a sign of reversibility. I'm going to use a proper example to illustrate this. Now, In the glycolytic pathway, there is a point at um, the point where glyceraldehyde 3-phosphate is interconverted to dihydroacetone phosphate, right? This P here stands for phosphate. This reaction is catalyzed by a triose isomerase enzyme. Isomerase enzyme in the glycolytic pathway. You're going to study this more when you're talking about glycolysis. Glycolysis is a very vast and ambiguous topic, but I just want to use this to illustrate something here. Now, depending on the need of the body, this particular intermediate in the glycolytic pathway can be converted to dihydroacetone phosphate, which is another intermediate of the pathway. Now, dihydroacetone phosphate can also be converted to glyceraldehyde 3-phosphate by the action of the triose isomerase, which is the enzyme in this case. That goes to tell us that most of the reactions that are catalyzed by enzymes can be reversed. Now, depending on the type of reaction now, and also the enzyme involved in that case, we'll know the amount of products that will be formed, or the amount of substrates that will be gotten back, right? So the seventh characteristics of enzymes is that they are proteins in nature, with the exception of ribozymes. Yes, in fact, in most texts, they will tell you that enzymes are proteins, but that is not entirely true in the context of biochemistry, because not all enzymes are protein. In fact, I already stated one here, with the exception of ribozymes. Ribozymes are RNA molecules that are responsible for conversion. Like, for instance, there's this enzyme called peptidyl transferase ribozyme. This particular enzyme is responsible for the formation of proteins, as formation of peptide bond of proteins in the ribosome, and it is not a protein in itself, it is an RNA molecule, right? So the eighth characteristic here is that some enzymes are inactive and therefore require coenzymes to activate them, and I gave an example to illustrate this. Now, there is a special name given to enzymes when they are in their inactive state. We refer to those set of enzymes to be zymogens. So in case you see this word somewhere, just take note of it. Right? Zymogens are enzymes in their inactive states. They are enzymes. When I say enzymes, I'm also talking about proteins in their inactive states. Right? Now they require a coenzyme. A coenzyme or a cofactor, anyone can do the activation. In this case now, this is pepsinogen. This is pepsinogen, which is a zymogen. This is zymogen, and then it is being acted upon by dilute hydrochloric acid. This dilute hydrochloric acid is the coenzyme we're talking about. But in this case now, it is not a coenzyme, it is an inorganic ion, a cofactor, because under the the agents, we have coenzymes and we have cofactors, right? One is organic and the other one is inorganic. We'll talk about that very much later in the video. So now, upon addition of this compound, this particular compound is being activated, and then we have pepsin, which is an enzyme on its own. And there's something very peculiar about enzymes. If you look very well, when I'll be talking about the classes of enzymes, most of them are going to be ending with this suffix known as AES. ACE. So wherever in your life you see something ending with ACE, just know that it is an enzyme. As the suffix, know it is an enzyme. Although, if you look at this is pepsin, it is also an enzyme. We also have renin, we have ribozymes, and a whole lot of them. But most of the enzymes are going to be ending with ACE. So it is a very important factor also. That's just an additional knowledge for you. So these are the characteristics of enzymes. There are also some that were not listed here, but if you go through the app, you're going to see them. They are quite self-explanatory. Okay guys, this is going to be the end of our class for today, and I'm going to be seeing you guys in the next Learn Extra class. Thank you for your time and patience.